Nice. Thank you. Okay. Thank you, Professor Julio. Thank you, everyone who is here. My name is Davi Souza. I am a Brazilian student researcher. I'm currently based in Saudi Arabia, and it's a big pleasure to be here with you to present one of the projects that me, Lucas, and our colleagues from SEDS Northeast Group is developing for now. And I'm also a greenhouse specialist, and I have been researching together with Habitat Martin, Professor Julio, for almost 4 years. And during this period, I was able to meet the colleagues from SES Northeast Group, and since then, we are being able to develop some student projects to develop the Brazilian space sector. Now, I give the floor to my friend Lucas to really start our
Hello,
presentation.
everyone. My name is Lucas Gabriel. I'm a biology science teacher, research, and analog astronaut from Habitat Mars. David and I will present the research entitled Student Projects to Promote Research in Space Biology and Small Satellites in Brazil. Project developed by SET Northeast Group. So, what is SET Northeast? We are a chapter of SETS Brazil, which is a chapter of SETS Earth. SETS Northeast is a group of students developing projects focused on space science and sustainability. We are represent all members here of the group. We are the Brazilian representation in the international community, and we disseminate science using STEAM education, such as engineering, agriculture, biology, and astrophysics. We are formed by a multidisciplinary team with students from several areas. So, what is space biology? Space biology, it is a branch of biology that studies the effects of space on living organisms, as well as the search for life outside the Earth. One of the main goals of space biology is to understand how living systems are affected with the conditions found in space environments. It seeks to understand how gravity or microgravity affects the morphology and physiology of cells from the early development and across the growth cycles. Sorry. Or how life can grow in environments with conditions different from those found on our planet. The results of this type of experiment help us design future space missions that will take living organisms to the other planets or other location in space. Understand how life on our planet develops under extreme conditions is one of the steps to colonize other planets. About CubeSats. CubeSats are the class of research spacecraft called nanosatellites. They can be easily changed and can be used as research and educational tool, introducing students to space science, engineering, and space technology development. We are where the topics take into consideration for development of the project that David will present in the next slide.
Well, talking about the SeedSet project, the entitled SeedSet project, um, we realized that these two presented areas by Lucas was of utmost importance if we wanted to develop any project based on the background of the group. So integrating both of the biology aspect and also with systems, especially on CubeSat aspect, we were able to find a great motivation of a project called Space Beam Project. This is a project developed by scientists from universities in France and also in Canada, which they came up with this paper that we are using as a reference also, which is towards an autonomous plant growing model for CubeSat. This paper was presented by Dr. Trollifel and his group in 2020 during the International Astronautical Congress, the cyber edition of it. Since then, we were able to collect and gather all of the data that would be necessary for us to come up with a similar project here in Brazil. On the space biology aspect, we came up with this experiment that would be focused on the analysis of the behavior of a plant species. Especially here, we take it into account the use of microgreens, which we will be presenting with more details in the couple of next slides. This would be the behavior of these microgreens in microgravity. In addition, we will be assess the environmental changes associated into the microclimate inside of the CubeSat, but also how does the microgravity will affect this microclimate. Also, regarding the CubeSat, similarly to the Space Beam project, we came up with a CubeSat module for autonomous plant growth to access key biological mechanisms associated with growing food in extreme environments, such as the microgravity, that may be useful to support human food health in future exploratory missions, which is the case of Moon, Mars, and beyond, and also benefit life here on Earth. Next slide, please, Lucas. The mission overview. Considering the financial constraints, considering that we are only a student group, we realized that differently from the Space Beam project, which use a 3U CubeSat, we would be interesting for us to start with only 1U CubeSat. And basically on this requirement, this initial requirement, we came up with other necessary requirements for our project. So differently from the common aspect of any agriculture or plant cultivation experiment, we conduct. Oh, there is some interference here. Sorry. We conducted the different path of common of mostly of space agriculture experiments, because some of these experiments start with the plant requirements to then develop the system. But differently, we came up with the requirements imposed by the systems to then came up with a proper solution for plant growth. And as you can see here, we have a CubeSat which needs, for a 1U CubeSat, we need at least half U use for this subsystems, which is the power system, the data handling system of the CubeSat, and also the communication system. And then in the another half U of the CubeSat, we were able to put our payload inside of it, which is a cultivation chamber that we will be presenting in the next slide. Please, Lucas. Well, as you can see here, inside of this closed cultivation chamber, the payload overview is basically the biological experiment, the biological materials that we'll be using for our experiment, and also the system sides, the system aspect. On the biological side, we have here the microgreens. We also would have the nutritive solution to grow these microgreens, and also the substrate. On the system side, we came up with a solution using microcontrollers, especially the ESP32 microcontroller associated with a camera and sensors, a battery, and a bunch of sensors to collect data on radiation, CO2 removal, air quality, temperature and humidity, and also collect other important parameters on the plant, such as the light incidence inside of the plants, above the plants. Next slide, please, Lucas. To develop this experiment, we came up with three main materials for the agriculture aspect, for the biological aspect. The first one is the substrate, which we selected the use of phenolic foam. Then we will be able to integrate the phenolic foam with a hydro-retentive polymer, which is commonly used in agriculture to keep the root environment more humid for long periods of time. With that, we are able to integrate this polymer with the nutritive solution for the microgreen, and putting this inside of the CubeSat to avoid any liquid material inside of our experiment, which will potentially compromise or putting our CubeSat into a risk situation. This is a thing that we want to avoid. And related to the species of microgreen that we selected, we came up with the red cabbage, which is a well-documented species by NASA with some studies on microgreens that basically has a high amount of nutritive content, also have a short growth cycle, which varies from 7 to 10 days since after the germination phase, and also has a short size. Especially these microgreens can achieve at least 8 centimeters of height. And next slide, please. And the experiment overview, as I mentioned, we have the cultivation aspect of our system with the substrate, with the polymer, and also we would have this system side inside of the cultivation chamber with the systems for control and monitoring, together with some LED lightings to cultivate our crops. And some of the outcomes of it, we would have some pictures as data to have an idea of the physiological parameters of our experiment, especially to analyze the growth of the microgreens, and also sensors to gather necessary data on environmental parameters, for example, and visualize this, like the environmental response of the system in real time. Next slide, please. Finally, we are integrating both important topics here, which is space and sustainability, showing people the potential of our project and the collaborative engineering to deliver, develop, and verify a balanced solution for extreme environments. Our experiment especially will allow us to find possible applications of plant growth to extreme and harsh environments conditions on our planet, and also enable an approach to agriculture in environments that are not suitable to grow plants, such an example, semi-arid regions, arid regions, desert regions, and also space. And finally, we will be able to show the ability of space to ensure sustainability on Earth, and considering all of the student integration posed here that we have presented here to you, we are being able to inform, inspire, engage, and impact many students with the potential of the space development in our country. Next slide, please.
We have recently participated in the OPSAT, Brasilia Satellite Olympiad, where we got the 3rd place in the regional stage. But we did not finish the project in time to participate in the event as we wished. The work done is not yet finished, and we are working so that we can continue with the CITSET project. It was a great opportunity to show our space biology proposal to students from all over the country. And last, we thank you all for watching the presentation of our project. And if there are any doubt, we are up to discussion and partnerships in your personal contacts or in the set social networks. Thanks.